I want to start with a number today. 13 times 10 to the 28th.
It is a number that is difficult to hold in your head. It's not built for the human brain.
It's impossible to hold in your head. We always use the stars as our benchmark for infinity, right? We look up, we see the Milky Way, we're told there are billions and billions of stars, and we feel insignificant.
It's our go-to metaphor for unimaginable scale.
Exactly. But this number 13 times 10 to the 28th is the estimated number of bacteria found in the world's oceans. And that is, get this, 100 million times the number of stars in the known universe.
And that's just the bacteria. That's not even counting, the rest of the players in the water.
Right, and that's where the angry part comes in for me. It's this realization that the universe isn't just out there, it's right here, it's down here. And we are just walking through it, completely, utterly oblivious.
We are outnumbered to a degree that is, it's statistically absurd. And if you want to dial up that anxiety just a little bit, you have to look at the activity level, not just the population count.
Yeah.
The source material highlights a terrifying statistic about viruses in that same ocean water.
This is the war of attrition statistic. This one is just, it's relentless.
It's relentless, exactly. We are looking at approximately 1 times 10 to the 23rd viral infections occurring every single second in the ocean.
Or second.
Every single second. Those infections are wiping out between 20 and 40% of all bacterial cells. Every day.
So it's not a static population, it's a battlefield.
It is a constant, violent turnover of biomass that releases chemicals, carbon, nutrients, everything back into the water. It's a blender.
So the chemical makeup of our planet, the very air we breathe, the composition of the water, it's basically the byproduct of a microscopic war.
That is the perfect way to frame it. And that really is our mission for this deep dive. We're working through a comprehensive review of the fundamentals of microbiology. And the goal isn't just to look at germs that make you sick.
No, this is bigger.
We are decoding the invisible empire that dictates the physical reality of Earth.
And it's not just in the ocean. That's the other thing. It's intimate. It's on us. It's in us.
It is us in many, many ways. And to understand that, we need to distinguish between the truly living, the bacteria, the fungi, the protists, and the things that our sources call the undead agents.
The viruses and the prions? The zombies of the microscopic world.
Correct. The things that aren't quite alive, but are definitely not dead.
Before we get to the zombies, though, let's establish the scope here. When the text defines microbiology, it's being very specific about what counts. We're talking about single cells, cell clusters, and these non-cellular agents. But the impact of these tiny things completely belies their size. I think the biggest shock for me, and probably for anyone listening who, loves a walk in the forest, is what you could call the oxygen myth.
It's a classic misconception. It really is. We are taught from kindergarten that trees are the lungs of the earth. Save the rainforest, save the planet.
And you should. We're not saying don't save the rainforest.
No, of course not. That is absolutely true and important for biodiversity and countless other reasons. But if you look at the raw data on photosynthesis, the heavy lifting isn't being done by the big green things we can see.
It's the microbes.
It's the microorganisms. It's the algae, the cyanobacteria in the oceans. These are the engines carrying out the majority of photosynthesis on the planet. They are the ones cycling the essential elements that life depends on. Carbon, nitrogen, sulfur, hydrogen, and, yes, oxygen.
So if you scrubbed the planet of all the trees, it would be catastrophic, a disaster.
An ecological nightmare, yes.
But if you scrubbed the planet of microbes?
The biological cycles would collapse almost instantly. The planet's life support system would just shut down. You wouldn't have nitrogen fixation for plants to grow. You wouldn't have decomposition to recycle waste. The entire gear system of the biosphere is microbial. It would all just grind to a halt.
Okay, let's bring that scale down to the individual listener, because saying they run the planet is, it's a bit abstract. Saying they run you is a lot more confronting.
It is.
The source material breaks down the human biome, and frankly, it challenges the very definition of what a human actually is.
It's an identity crisis waiting to happen. If you look at a human body, you assume it is a single, unified organism. But biologically, you are a walking ecosystem, a community.
A crowded one.
Incredibly crowded. The estimates vary, of course, but generally 50 to 60% of the cells in your body are not human cells, they are microbial cells.
So by cell count, I am a majority shareholder in my own body, but barely, just barely over 51%.
Barely. You are essentially a vessel, a highly complex walking hotel. And to make that tangible, consider your gut. The bacteria residing in your digestive tract, your gut flora weigh about 1 kilogram in an average adult.
That's a bag of sugar. I'm carrying around a bag of sugar's weight in living bacteria.
And that's just the physical weight. The weight doesn't matter nearly as much as the information they carry.
The genetic information.
Exactly. We place so much importance on the Human Genome Project, right? We spend billions mapping our DNA. We think our DNA is the blueprint of destiny.
It's the code of us.
Right. But the number of genes contained within that gut flora outnumbers the genes in your own human genome by 150-fold.
150 times. This is the part I'm trying to wrap my head around. We know that genes code for proteins, and proteins do the work. They send signals, they digest food, they regulate mood, they do everything.
They are the machinery.
So if there are 150 times more microbial genes than human genes in my body, who is actually driving the bus?
That is the multi-billion-dollar question researchers are asking right now. We know these microbial signals affect everything from your immune system to your serotonin levels, which affects your mood. You are constantly bathed in a chemical soup created by that 1 kilogram of bacteria.
There was one other statistic in this section that I found. Well, vivid is the word. It's about waste.
Yes. The text points out that a human adult will excrete his or her own body weight in fecal bacteria every single year.
It just really drives home the factory aspect of our bodies. We're basically bioreactors constantly producing and expelling this microbial mass.
We are, but if the bacteria are the workers in the factory, we also have to talk about the ghosts in the machine. We mentioned the undead earlier, the viral components.
The viral ghost in the machine. This part of the notes actually gave me a bit of a chill. We tend to think of viruses as invaders. You catch a cold, you fight it off, it's gone. But the text says that's not the whole story. Not even close.
It's not. It turns out about 8% of the human genome, the DNA that you think of as yours, that you inherit from your parents, is actually derived from remnants of viral genomes.
So 8% of my personal blueprint is just old, leftover viral code.
Precisely. These are called endogenous retroviruses. Millions and millions of years ago, these viruses infected our primate ancestors. But instead of killing the cell or just replicating and leaving, they managed to do something tricky.
What's that?
They inserted their genetic code into the germline, the sperm or the egg cells.
And once it's in the germline, it gets passed down. It's inherited.
It gets inherited generation after generation after generation. It becomes a fossil record of ancient pandemics embedded in your own chromosomes.
So our DNA is like an archaeological big site for ancient diseases.
It is. And some of those viral genes have even been co-opted or domesticated by our own bodies. There's strong evidence that the development of the placenta in mammals, the very thing that allows us to have live births, relies on a protein that was originally viral.
That is wild. We are, in a very real sense, part virus. It just blurs the line of identity completely. It really supports this idea mentioned in the introduction that microbiology is the arena where biological diversity is truly understood.
It is. In fields like zoology or botany, you're studying the macro world. You see the results of evolution. But in microbiology, this is where the rules of life were written and where they are constantly being rewritten. The text calls it the place where originality can be fully met.
What does that mean in a practical scientific sense? Originality.
It refers to the speed of inquiry. The scientific method relies on testing hypotheses. If you are studying, say, elephants, testing a hypothesis about their evolution might take you 300 years because their generation time is so long.
Right.
You can't see evolution happen in real time.
But with microbes,
they can reproduce in minutes, 20 minutes for an E. coli. You can see thousands of generations in a week. You can literally watch evolution happen in a test tube. You can test a hypothesis and get an answer. That's why biochemistry, molecular biology, and genetics all converge on microbiology. It's the high-speed laboratory for understanding life itself.
So we've established that we are covered in these things, filled with them, and that they are evolving at this incredible blistering pace. The natural assumption is that this is a war zone. I mean, is my immune system versus the world. But the text emphasizes that it's not just a cage match.
No, war is just one setting, one possible outcome. The broader concept is symbiosis.
Which I think people misuse all the time. I hear people say we have a symbiotic relationship when they just mean we get along. But scientifically, it just means living together, right?
Yeah.
It doesn't imply that it's good or bad.
Correct. Symbiosis is defined as a continuing association of different organisms. That's it. It's a spectrum. It ranges from we're best friends and can't live without each other all the way to I am going to eat you from the inside out.
Okay, let's start with the best friends end of the spectrum then. Mutualism.
Mutualism is where both parties benefit. The classic textbook example, the one everyone learns, is lichens. You've seen them, those crusty, leafy, colorful growths on rocks or tree bark.
I always just assumed that was a type of moss
Mm-hmm.
or, some kind of single weird plant.
Most people do. But a lichen isn't one organism, it's a partnership. It's a marriage between a fungus and a phototroph.
A phototroph being the light eater, an organism that does photosynthesis.
Right, usually an alga or a cyanobacterium. The deal they strike is really elegant. The phototrope is the primary producer. It uses photosynthesis to create sugars and food for both of them.
So it's the chef.
It's the chef. The fungus, which can't make its own food, provides the structure. It builds the house, the structure called the thallus, which anchors them to the rock and, crucially, protects the delicate alga from drying out or from too much UV radiation.
So the fungus provides the real estate and the security system, and the alga provides the catering.
Exactly, and it's an obligate relationship in many cases. If you separated them, they would die in that harsh environment. Together, they can colonize bare rock where nothing else could survive.
But then we slide down that spectrum to the relationships we're, more afraid of. Parasitism.
The dynamic where one organism, the parasite, benefits at the expense of the other, the host. This is the realm of pathogens, disease-causing bacteria, and viruses. But there is a fascinating nuance here that the text brings up regarding how we study them.
The mimicry problem.
Yes. Listeners might wonder, if we know a certain bacteria causes a disease, why don't we just grow a bunch of it in a lab and figure out how to kill it?
It seems straightforward enough.
The problem is, a parasite has evolved over millions of years to live specifically inside a host. It relies on the host's exact temperature, the exact pH, the precise mix of nutrients that the host provides.
It's like a pampered guest. If the pillow isn't fluffed exactly right, it refuses to check in.
Or it just dies. It refuses to grow. To study a parasite in a lab, you have to achieve effective mimicry of the host environment in a petri dish or a test tube. If you can't fake that environment perfectly, the organism won't cooperate, it won't reveal its secrets. This is why some diseases are still so hard to understand. We simply haven't figured out how to keep the culprit alive outside the body.
And all of this, the lichens, the parasites, our own gut bacteria, it's all the result of billions of years of trial and error. It's evolution.
Natural selection acting on genetically diverse organisms. Which brings us to the biggest split in the biological world. The Great Divide, as the text calls it.
This is the eukaryote versus prokaryote split. I remember these terms from high school biology, but I feel like I need a refresher on why this distinction matters so much. It sounds like jargon, but it's fundamental.
It matters because it's about architecture. It's the difference between a studio apartment and a mansion with a bunch of different rooms.
Okay, walk me through the mansion first. That sounds more interesting.
The mansion is the eukaryote. You are a eukaryote. So are plants, animals, fungi, and protozoa. The defining feature is the nucleus. Eukaryotes have a membrane-bound vault, a separate room, where they keep their precious DNA. It's physically separated from the rest of the cell's chaos.
The main office or the vault?
Exactly. And they also have other specialized rooms, organelles. They have mitochondria, which are the power plants. They have the Golgi apparatus for packaging and shipping proteins. It's all highly compartmentalized and complex.
And the studio apartment?
That's the prokaryote. This includes the bacteria and the archaea. No nucleus, no wall separating the DNA from the rest of the cell. The genetic material is just sort of floating there in the main living space, the cytoplasm, in a region we call the nucleoid.
So no separate rooms. Everything's happening in one open plan space.
Right, and they generally lack those complex membrane-bound organelles. It's a much simpler, more streamlined design.
It sounds primitive.
Primitive is a loaded word. I prefer to think of it as efficient. They are streamlined for speed, they can reproduce much faster and adapt quicker because they aren't bogged down by all that complex internal architecture. They are the lean startups of the biological world.
Okay, so that covers the living. We have the mansion dwellers, us, and the studio apartment dwellers, bacteria. But now we have to enter the twilight zone. Section 3 of our deep dive. The non-living agents.
The things that exist right on the edge of what we even define as life.
Viruses. The source material is very, very clear. They are not considered living organisms. But having just recovered from a flu recently, I can tell you they feel pretty active. Why don't they get the life badge?
It comes down to independence. To be considered alive, a cell generally needs to be able to sustain itself and replicate on its own. It needs metabolism. A virus cannot do that. A virus particle sitting on this table is just a complex chemical. It's inert. It's a rock.
It does nothing.
Absolutely nothing. It has no metabolism. It doesn't consume energy. It doesn't grow. It only wakes up when it comes into contact with a suitable host cell.
So it's purely potential energy, like a loaded mousetrap waiting for a mouse.
That's a great analogy. And they are incredibly picky mousetraps. This is a concept called tropism. Viruses are usually restricted to infecting specific hosts and even specific cell types within that host.
Right, so a plant virus generally isn't going to infect me.
It'll just bounce right off. And a virus that targets your liver-like hepatitis will completely ignore the cells in your nose. They're looking for a very specific lock, a receptor on the cell surface to put their key into.
But the text mentions a discovery that sort of breaks this whole hierarchy we imagine. Virophages.
This is one of my favorite recent developments in microbiology because it adds a whole new layer of complexity. We used to think in a simple chain. Big things eat small things. Bacteria infect humans. Viruses infect bacteria.
A clear food chain of infection.
A clear chain. But virophages are viruses that infect other viruses.
That feels like cosmic justice. Even the viruses have to worry about getting sick.
It's not just justice, it's complexity. It implies an infection web that goes much deeper than we thought. You might have a massive virus, for instance, that infects an amoeba, and then a tiny virophage comes along and infects that massive virus, hijacking its hijacking attempt.
A pirate pirating another pirate.
It's a perfect description.
Let's look at the mechanics of that hijack. Because viruses are terrifyingly efficient. What are they actually made of? What's in the box?
Pure minimalism. A typical virus, like an adenovirus, is tiny, about 90 nanometers. It consists of nucleic acid, its genetic code, which can be either DNA or RNA wrapped in a protein coat called a capsid.
And that's it?
That's basically it. Some have an extra lipid envelope that they steal from the host cell on their way out, but essentially it's just a code in a box, a message with a delivery system.
And a delivery is brutal.
It's a hostile takeover. The virus attaches to the correct cell. It injects its code. That viral code then completely redirects the host's enzymatic machinery. It walks into the factory, fires the manager, and puts its own blueprints on the assembly line.
Stop making new skin cells. Stop doing your day job. Start making thousands of copies of me.
Exactly. And in some cases, the viral information is actually incorporated directly into the host's own DNA. This is what's called a provirus. It's a sleeper agent.
It hides in plain sight.
It can sit there, hidden in your own DNA, being copied every time your cell divides, for years before something triggers it to activate.
And when it activates?
Maturation. The cell's machinery starts to assemble thousands of new viral units based on the provirus code. And then usually the cell lyses or bursts. It dies, releasing this new swarm to go find new victims.
Okay, I get viruses. They are code thieves. They are information pirates. But the next one on the list, this is the one that really scares me. Prions.
Prions are in a league of their own. If viruses are code in a box, prions are just the box, a twisted, broken box.
They are proteins, just proteins. No DNA, no RNA, no genetic code whatsoever.
None. Zero.
So how can they be infectious? The central dogma of biology, the thing we all learn, is that DNA makes RNA, which makes protein. Information flows in one direction. How does a protein replicate itself without any instructions?
That is the mystery that baffled scientists for decades and won a Nobel Prize. Prions are the zombie proteins. Here's how it works. We all have normal, healthy prion proteins, we can call them NP, in our brains. They have a specific folded shape, and they do a job we don't fully understand yet.
Okay, they're supposed to be there.
They're supposed to be there. But an infectious prion is an abnormal prion protein, or PP. It's the exact same chain of amino acids, but it has been misfolded into a different, twisted, stable, and dangerous shape.
Okay, so a normal protein and a corrupted one.
Exactly. Now, here's the terrifying part. When this twisted abnormal protein touches a normal protein, it acts as a template. It physically forces the normal protein to snap and refold into the abnormal shape.
Kiss of death. It corrupts it.
It's a chain reaction. One corrupts one, now you have two, those two corrupt two more, now you have four. Four touch eight. It's an exponential cascade of misfolding. These misfolded proteins are very sticky, they clump together, and they literally turn the brain tissue into a sponge.
Hence the name. Transmissible spongiform encephalopathies.
Diseases that are infectious and turn the brain into a sponge. In humans, this shows up as Kuru, which was spread by cannibalism, or Creutzfeldt-Jakob disease, CJD, or the one that is the stuff of nightmares. fatal familial insomnia.
Which is exactly what it sounds like.
You lose the ability to sleep permanently until you die from exhaustion and brain degradation. In animals, we see it as mad cow disease or scrapie in sheep or chronic wasting disease in deer and elk.
And the resilience of these things is the kicker. You can't just cook them out of infected meat.
No, they are incredibly resistant to heat, radiation, and proteases, the enzymes our body uses to break down old proteins. Standard sterilization often fails. There are documented cases of CJD transmission through surgical instruments that were sterilized, but still carried the infectious prion.
Note to listener, do not eat brains or spinal tissue.
A solid life rule to live by.
Just to round out the non-living agents, there was a quick mention in the text of viroids.
Yes. They're another fascinating example of minimalism. If prions are just protein and viruses are protein plus code, viroids are just the code. Naked RNA molecules, no protein coat, just floating around.
And they do what?
They mostly infect plants, causing things like potato spindle tuber disease. They get into the cell and use the cell's machinery to make copies of their RNA. It just goes to show that nature will try every possible combination of molecules to see what survives and replicates.
All right, let's scrub up and get back to the world of the living. Section 4, the prokaryotes, the bacteria and the archaea. We talked about their studio apartment structure, but I want to dive into their capabilities, because despite being architecturally simple, they are metabolic wizards.
They can do things biochemically that we can't even dream of. We humans are, metabolically speaking, very boring. We need oxygen, we need organic food. That's it. Prokaryotes. They have figured out how to occupy every conceivable niche on Earth.
Give me an example of the weird ones. The stuff that's not just E. coli.
Okay, look at purple bacteria. They perform photosynthesis, they convert light to energy, but they do it without producing oxygen as a byproduct. They use different chemical pathways. Or look at the true anaerobes. These are organisms that find oxygen to be a deadly poison. They have to live in places with no air.
And they breathe other things. How does that work?
Well, respiration is just a process of moving electrons around to generate energy. We use oxygen as the final dump, the final electron acceptor. These bacteria can use other things. They can breathe sulfur or nitrogen compounds or even iron.
They breathe rust.
Essentially, yes. And then you have fermentation.
This one we're more familiar with.
We are, or at least with its products. Fermentation is a way to get energy from just rearranging chemical substrates without any external electron acceptor at all. It's inefficient compared to oxygen respiration, but it works in a pinch. And luckily for us, the waste byproducts are things like lactic acid and alcohol, yogurt, cheese, beer, wine, kimchi, all thanks to bacterial fermentation.
I want to talk about their social lives. Because again, our mental image of bacteria is of these lonely drifters just floating around. But the text introduces the concept of quorum sensing. This blew my mind.
It's bacterial communication. It completely challenges the idea that bacteria are just mindless individual cells.
But how does it work? How do they talk to each other?
Bacteria are constantly at a very low level releasing small chemical signal molecules into their environment. These are called autoinducers. You can think of it as a pheromone.
Okay.
When there are only a few bacteria around, that signal just drifts away. The concentration is too low to notice.
That's like whispering in a hurricane. Does anyone out there know?
Exactly. But as the population grows, as they divide and divide, the concentration of that chemical signal rises. When it hits a specific threshold, that's the quorum, it binds to receptors and triggers a genetic switch in all the bacteria simultaneously.
So they count themselves? They take a census of their own population density.
They take a census, and once they know they have the numbers, they act as a group. They might all turn on a gene to produce a toxin at the same time. This is tactical.
Why is it tactical?
Think about it. If one single bacterium releases a toxin in a human body, your immune system squashes it instantly. It's a non-event. But if a billion bacteria wait until they are ready and then they all release the toxin at once.
Overwhelmed defenses. Coordinated attack.
Precisely. They can also use it to form biofilms, those slimy protective layers you find on unbrushed teeth or on catheters or on rocks in a stream. Quorum sensing allows single independent cells to act with the coordinated behavior of a multicellular organism.
They're also trading information, not just sending signals. We have to talk about plasmids. The source material makes them sound like the trading cards of the bacterial world.
Plasmids are absolute game changers in evolution. In a eukaryote like us, our DNA is locked away in the vault. It's one big package. In bacteria, they have their main chromosome, but they also often have these tiny, separate, circular loops of DNA called plasmids, and they can replicate independently of the main chromosome.
And they can just hand them off to other bacteria.
Yes. Through a process called conjugation, one bacterium can build a little bridge, a pilus, to another and pass a copy of its plasmid through. And these plasmids often carry specialized, non-essential, but very useful genes. Superpowers, if you will.
Like antibiotic resistance.
That's the most famous and dangerous example. A completely harmless soil bacteria might have a plasmid that gives it resistance to, say, penicillin. It just happens to meet a dangerous pathogen like Staphylococcus. It passes the plasmid over. Now suddenly the staph is resistant to penicillin.
And the text says they have a broad host range. What does that mean?
That's the really scary part. They don't just trade plasmids within their own species. They can trade across very diverse species. It's an open-source biological weapons market. This is why antibiotic resistance can spread so terrifyingly fast through hospitals. Bacteria are actively sharing the genetic codes to defeat our best drugs.
Moving into Section 5, Evolution. This brings us to the origin story of our own complex cells, a theory called endosymbiosis. We mentioned earlier that we are complex ecosystems. But this theory suggests that even our own human cells are actually just ancient bacterial communes.
This is the leading theory for how complex life, eukaryotic life, began. And the evidence is really compelling. If you look inside your cells at the mitochondria, the little power plants that generate your energy, they look suspiciously like bacteria. They're the same size and shape. They have their own DNA.
Which is weird, right? Why would a part of a cell, an organelle, need its own separate set of blueprints?
Unless it used to be a separate organism. The theory is that billions of years ago, a large predatory cell engulfed a smaller bacterium. But for some reason, instead of digesting it, something happened. They struck a deal. A symbiosis formed.
I won't eat you, and in return, you make energy for me.
And I'll protect you from the harsh outside world and bring you food. Over billions of years of coevolution, that little bacterium became the mitochondrion we know today. It lost its ability to live on its own. It became domesticated. The same thing is thought to be true for chloroplasts in plants.
So we are literally powered by enslaved ancient bacteria.
Or you could say, we are the partnership. We wouldn't exist without them. This domestication also happens in the other direction. Evolution can lead to a loss of function in parasites.
The text mentioned mycoplasmas as an example.
Right. Mycoplasmas are a type of bacteria that, at some point in their evolution, decided to stop building walls. They lost the ability to form a cell wall, which is a major component for most bacteria. To survive, they have to steal cholesterol from their host cells to stabilize their membranes. They became dependent thieves.
And chlamydia and rickettsy are even more extreme.
They are obligate intracellular parasites. They've gone so far down the parasitic path that they've lost the ability to make their own essential metabolites like ATP, their energy currency. They can only survive and reproduce inside another living cell. They evolved into total dependency.
Now, with all this swapping of genes and losing of functions, how on earth do we classify them? The text makes a good point that just looking under a microscope isn't enough anymore. A dot looks like a dot. A spiral looks like a spiral.
For a long time, that's all we had. We'd rely on visual shape or the Gram stain, a chemical test that dyes bacteria purple or pink depending on the structure of their cell wall. It's still a very useful first step. But the real revolution is phylogenetics.
Reading the DNA and comparing the sequences.
Exactly. Comparing the sequences of fundamental genes, like the genes for ribosomal RNA. And this genetic analysis led to the biggest surprise in modern biology, the discovery of the archaea.
For the longest time, we thought these were just weird bacteria, right? They're lumped in.
We called them archaebacteria. We found them living in boiling hot springs or incredibly salty water or pools of acid. We thought they were just bacteria that liked extreme environments. But when Carl Woese and others started looking at their DNA sequences, they realized they weren't bacteria at all.
They are a completely separate third domain of life, as different from bacteria as we are.
And here is the real twist. Eukaryotes, that's us, plants, fungi, we're more closely related to these strange archaea than we are to regular bacteria like E. coli.
So my long-lost cousin is the weird organism living in a sulfur vent at the bottom of the ocean.
Genetically speaking, yes. Archaea have certain traits, like having introns, little non-coding interruptions in their DNA code. that bacteria don't have, but we eukaryotes do. It suggests that the original host cell that started the whole eukaryote lineage might have been an archaean, which then engulfed a bacterium that became the mitochondrion.
It's a messy, complicated family tree.
Evolution is always messy. It's not a clean ladder. It's a tangled bush.
Let's finish up our tour of life with Section 6, the Eukaryotic Zoo. This covers the protists and fungi. Protist seems like a junk drawer category, a place to put things that don't fit elsewhere.
It is a catch-all term. It's what biologists call polyphyletic, which is a fancy way of saying these things come from different evolutionary lines, but we lump them together because they're small, eukaryotic, and don't fit into the plant, animal, or fungi kingdoms. It traditionally includes algae, protozoa, and slime molds.
Okay, let's start with algae. We mentioned earlier they produce a lot of our oxygen, but they can also be deadly. The text brings up red tides.
This is a phenomenon caused by a massive bloom of dinoflagellates, a type of unicellular algae. When the conditions are just right, warm water, lots of nutrients, they can reproduce explosively, turning the water a reddish-brown color.
And some of them produce toxins.
Very potent neurotoxins like saxitoxin, and this enters the food chain.
Ow.
Filter-feeding shellfish like oysters, mussels, and clams eat the algae. The shellfish are immune to the toxin, but they concentrate it in their flesh to incredibly high levels. Then a human, or a seabird, comes along and eats the oyster. The result is paralytic shellfish poisoning. It shuts down your nervous system. It can be fatal.
And there's even an alga that can infect pets. That seems very strange.
Prototheca. It's a fascinating case of reverse evolution. It's an alga that lost its chlorophyll. It can't photosynthesize anymore, so it had to find a new way to live. It turned into a pathogen. It causes nasty skin and systemic diseases in dogs, cats, and cattle.
Then we have the protozoa, the hunters of the microbial world.
The single-celled animals, essentially.
Yeah.
They don't use photosynthesis. They eat, and they do it through phagocytosis. They literally wrap their flexible bodies around their prey like a bacterium and engulf it.
Using false feet.
Pseudopodia, that's what amoebas do. They ooze along, extend these pseudopods, wrap around a bacteria, and pull it inside into a food vacuole. Others, like the ciliates, are covered in tiny hairs that they beat to create a current sweeping food particles into a designated mouth called a cytostome. It's a microscopic jungle down there with predators and prey.
And finally, the fungi. This is where we find the biggest organism on our list today, which is kind of shocking.
Fungi are fascinating. They aren't plants. They don't use light. They aren't animals, though they're more closely related to us than to plants. They're decomposers. They grow as a network of fine branching filaments called hyphae. The entire interconnected mass is called a mycelium.
And the record holder is an organ.
The humongous fungus. It's a single mycelial network of a honey mushroom species in the Malheur National Forest. It covers 2,400 acres. That's nearly 10 square kilometers.
And that's one single organism.
One continuous genetic individual. It's estimated to be thousands of years old. It's living mostly underground, spreading through the soil and eating the roots of trees. It is arguably the largest living thing on planet Earth by area.
Fungi also have this ability to shapeshift, which the text highlights as being really important for disease, the dimorphic fungi.
Yes, this is a major issue in medicine. Many pathogenic fungi, like the one that causes histoplasmosis, exist as a mold in the environment, growing those long, thread-like filaments. But when you inhale their spores and they enter the warm 37 degrees Celsius environment of the human body, they switch their growth pattern. They turn into yeast single budding cells.
It's a tactical camouflage. Why do they do that?
It allows them to travel through the bloodstream more easily and evade certain parts of the immune system. It's a distinct survival mechanism that is regulated by temperature. Mold in the cold, yeast in the heat.
We have to mention the slime molds before we wrap up, because they sound like something out of a 1950s sci-fi movie.
They are genuinely bizarre. A true slime mold forms a structure called a plasmodium. It's a giant, single-celled blob of cytoplasm containing millions of nuclei, but with no cell wall separating them. It's one giant, multi-brain amoeboid cell.
And it moves.
It flows. It pulses and creeps across the forest floor, a sheet of living goo searching for bacteria and other microbes to eat. But the genius is in its life cycle. When food runs out in one area, this blob doesn't just die. It stops flowing. It differentiates. Parts of it form a stalk. Other parts form a fruiting body on top, which then releases spores.
So it transforms from a single, giant hunting animal into a plant-like structure in order to reproduce and spread to a new location.
It shows the incredible flexibility of life. It's not locked into one mode of being. It's a beautiful illustration of the interdependence of living things and the evolution of specialized cells from a single, unified mass.
So we've covered a lot of ground today, from the 10 to the 28th bacteria in the ocean to the viral ghosts in our DNA, from the zombie proteins to the giant fungus in Oregon. If you had to synthesize all of this for the listener, what is the big unifying takeaway?
I think the takeaway is that the boundaries we draw are artificial. They're for our own convenience. We draw a line between living and non-living, but viruses and prions dance right on that line, challenging the definition. We draw a line between self and other, but our own bodies are riddled with foreign DNA and are utterly dependent on ancient bacterial symbionts.
So we aren't really individuals, we are collectives.
We are ecosystems. We are walking, talking vessels for a microbial world that is older, more diverse, and ultimately more resilient than we will ever be.
That is both a humbling and a slightly terrifying thought to end on. I want to leave the listener with one final provocation, something I scribbled down while reading about those virophages.
Go for it.
If 8% of our DNA is viral, and our energy comes from domesticated bacteria, and viruses can get sick from other viruses, is there actually a bottom to this? Or is it just Russian nesting dolls of infection and symbiosis all the way down?
That is the question that keeps microbiologists employed. Every single time we think we've found the floor, we find a trapdoor that leads to a whole new basement we never knew existed.
Look at the world with the new perspective today. It's a lot more crowded down there than you think. Thanks for listening to The Deep Dive.
Stay curious.